Welcome to the first seminar of the PacVec Spring Series for 2025. I think there's quite a few people who have joined us, so I'm going to get ready for our first speaker is Aine Blankenship-Cutshall. Aine is at the University of the Pacific as a graduate student, and she is, I understand, a UC Davis alumna. At the University of the Pacific, she is conducting graduate research examining the state of target site mutation resistance in California in Culex tarsalis populations with a partner, Eric Moyoung, under the guidance of Dr. Tara Tiemann, so she's in the Tiemann lab. She also serves as a laboratory technician at the Sacramento YOLO Mosquito and Vector Control District, where she conducts field surveillance of local mosquito and tick populations. Aine's research interests include vector-borne disease, the intersection of veterinary medicine and human public health, and the impact of public outreach on disease prevalence. So welcome, Aine. And if you have any questions, there will be time at the end of Aine's presentation to ask those. Or if you'd like, you can also post them in the chat during the talk or at the end of the talk, and we'll try and make sure that we address all of those questions. So, Aine, take it away.
Hi, thank you. I am super excited to be the first speaker in the spring seminar series. And like Neil mentioned, I'll be talking about the state of knockdown resistance in California's mosquitoes. I'm going to breeze through the background because this is a pretty knowledgeable audience. So as we know, West Nile virus is the most prevalent vector-borne disease in California. In 2024, 112 human cases were reported across 23 California counties. A pretty important vector of West Nile is Culex tarsalis. Culex tarsalis also vectors Western equine encephalitis and St. Louis encephalitis. This mosquito is so important because not only is it a local population, but it is common within both agricultural and residential areas, which puts it at a pretty unique intersection for vector-borne disease. Culex tarsalis faces insecticide pressure due to agricultural pesticide usage when it's in those agricultural areas, as well as vector control efforts and unrelated structural and industrial pesticide application. There are two main groups of insecticides we use to battle mosquitoes today. Those are pyrethroids and organophosphates. And for the rest of today's talk, we'll be focusing on pyrethroids. Pyrethroids work by binding to the insect nervous system, specifically the voltage-gated sodium channel. They work by preventing the closure of gates, which prevents axon repolarization, which leads to excitation and eventual exhaustion and depletion of the insect's nervous system. Insecticide resistance comes in many mechanisms, but we're going to focus on what I like to call kind of the innermost mechanism, which is target site resistance. That is conformational changes at the actual place where the insecticide is attempting to bind. This mutation is known as knockdown resistance. It is called L1014F. All that means is that the wild type is leucine, and there is a conformational change when the mutation to phenylalanine takes place. On the left here is just a schematic of the voltage-gated sodium channel, and you can see that this is a 4-domain, 6-subsection each 4, and on the 6th subsection of the 2nd domain, there is a hinge. When this hinge is open, that is when pyrethroids can bind. However, this mutation promotes closed state inactivation repolarization so that that hinge never opens and pyrethroids cannot bind. There's a secondary mutation at this site, which involves the changing of leucine to a serine. This does not confer resistance at the same rates as the aforementioned phenylalanine mutation, and both of them we see have a cost of survival. That is that closed state inactivation leads to weaker depolarizations than normal repolarization. So what's going on with Culex tarsalis and KDR resistance, as you've seen previously? What we saw in the Tiemann lab is that mosquitoes from mixed pools from both Sutter, Yolo counties, and the following year again in Sutter County saw that 85% of total alleles within the pool were phenylalanine. And that's pretty concerning, which brings us to this study and what we were trying to achieve. So when we were selecting sites for this study, we wanted to take a cross-California look at the state of insecticide resistance. So we have some sites in Northern and Central California and some more sites in Southern California. Moving from the top down, that's Lake County, Sutter County, Yolo County, Riverside, and Kern. With each mosquito, we asked vector control districts to send them over to us, and they were sexed and sorted by them. We then dissected them to get rid of any contaminant DNA that we did not want, and then purified that DNA to carry out genotype determination, which we used the cyber green fluorescence assay developed previously by Bridget Hughes in 2017. This assay differentiates alleles by milk curve analysis, influenced by the length of the GC-rich tail that we included in the primer design. That's a lot of words to just say we were influencing the milk curve of these different genotypes. This is a graph of what you might see when you do this type of assay, where along that bottom axis, the different genotypes are being influenced to melt and create peaks at different temperatures so that you can visually tease them apart a bit. So what did we find? It wasn't good. When we looked at modern populations in Northern California, this is the graph that's on the right here. You can see that this dark pink giant bar in every single county is phenylalanine. This is not genotypes, but instead allelic pools from each set of mosquitoes. And when we look in our Northern California mosquitoes, you see very low occurrences of the wild type allele, that's that light pink. And in Sutter Yuba, you saw no wild type alleles. I would also like to point out these small purple bars. That is the presence of that secondary mutation I mentioned earlier, serine. In Southern California, there's kind of a different story going on. In Kern, it looks very much like Northern California. High occurrences of that primary phenylalanine mutant and low occurrences of that purple serine. And Coachella Valley, however, there's a very different story going on. We see that there is a little over half of that wild type allele still present in the population. However, this does not mean that this population is not still resistant, and we'll get into that a little bit more as we talk about genotypes. So each mosquito, we not only considered the allelic frequencies of the entire population, but the frequencies of the genotypes of the mosquitoes within the population, because these genotypes are a little bit more what translate to phenotypic resistance. So what we saw here is that we're back to Northern California, and there are 3 counties. On the left of each county is a historic population from 2007, and on the right of each county is the modern population. What I want you to notice is that we go from these big green bars, those are homozygous wild type mosquitoes, to dark pink bars, which are homozygous phenylalanine mutants. I also want to point out the presence of those little purple bars. Those are actually phenylalanine serine, what I will refer to as double mutants for the rest of this talk. And their presence is pretty interesting. In Southern California, we again see very different stories. I want to focus on Kern County first because the modern population looks a lot like the modern populations of other counties. However, we see that in 2007, we started off a lot worse than in other counties. That big dark pink bar is still homozygous phenylalanine mutants. And there's a very low occurrence of wild type mosquitoes in Kern County, all the way back in 2007. In Coachella Valley, we have yet again a different story. There is a large presence in 2007 of wild type alleles and already some presence of phenylalanine homozygous. As we move to modern day, we see that there are much fewer wild type homozygotes, but they still persist in this population, which is unique amongst our 5 counties. And there is a very large instance of leucine, that's our wild type, and phenylalanine, our primary mutant, heterozygotes. One reason that this might be, besides these just giant geographic differences, is insecticide use. Insecticide use is probably the primary pressure that these populations face that would influence the changes in allelic frequency. So on this graph here, I have the pounds of pyrethroid applied in each county over a 30-year time period, normalized by acreage, as some of these counties are much larger than the others. I also have these sorted by kind of who sprayed the insecticides. That is, red is agriculture, green is industry, teal is public health, and purple is structural pest control. And what you can see here is that pyrethroids have been used pretty steadily in every single county throughout the study period. And I want to point out that agriculture, that's that red, and structural, that's purple, are the kind of two major players in most counties. So what does all of this mean? Insecticide use in every single county is definitely our major player. In most counties, pyrethroid application increased somewhere between our 2 time points. I've highlighted that in 4 of the counties here. And you can see that between our 2 time points, there's some sort of peak. And during that peak, there was potentially very strong selective pressure. When looking at who was spraying, we also need to consider that, because not all of these industries were targeting mosquitoes. In red here is agriculture, and that is potentially non-target exposure of insecticides to these mosquitoes. Farmers are unlikely to be spraying against mosquitoes. They're probably spraying for agricultural pests. However, we know that Culex tursalis is present in these agricultural areas. So even though they were not the target of these sprays, they were likely exposed to and therefore faced selective pressure because of this agricultural spraying. Moving into some of our other players here, I want to highlight that in purple, we have structural pest control being a pretty key player in a couple of the counties, particularly in Sacramento County, as well as industry in Sacramento County. These contain both potential target and non-target exposures, as companies like Fork and Clark and other private pest control companies do advertise their services for mosquito control. So there are potential target applications there, but there are also lots of non-target applications here, as these companies are also called out for there are too many gnats in my yard and stuff like that. What I want to point out, though, with these non-target exposures, both from these and agriculture, is that mosquitoes may not only face selective pressure during the sprays themselves, but also possibly selective pressure due to residual insecticide. In 2010, Weston and Lively found that there was bifenthrin in the American River. That's a river here in Sacramento that was found at levels toxic to amphipods. That's what they were testing for specifically. And so I just say this to point out that there is definitely evidence for residual insecticide in the environment that may affect mosquito populations. And so all of these selective pressures, of course, result in population changes. On this right down here, we have, once again, the allelic frequencies, not the genotypic frequencies, across all of our counties, with our historic populations on top and our modern populations on the bottom. And what we can see is that from 2007 to 2021, these wild type alleles have all but disappeared. On the top, we have really large, big, light pink bars, with the exception of Kern County. And on the bottom, with the exception of Coachella Valley, those pink bars have all but disappeared, which has concerning implications. These concerning implications are, of course, not isolated to Culex tursalis. There is another prevalent Culex species across these areas, 2 others, Culex pipiens and Culex kingfishitis. And in Northern California, it's Culex pipiens. And in 2020, Yoshimizu reported that the frequency of resistant KDR genotypes, those are these same mutations, had increased 1.5-fold in 2014 when comparing that to the same population sampled in 2012 by a previous study. And what we saw here was that 72% of the alleles tested in 2014 Placer County Culex pipiens were resistant alleles. And Placer County is very close to some of our Northern California populations. And both of these things put together with our findings have pretty concerning implications for the successful control of the Culex species complex, which isn't great because those are our main vectors here in California. I also want to talk about the resistance of individual mosquitoes, and here I have genotypes up again. Individuals containing phenylalanine alleles are more likely to have some sort of resistance. That's why we call it a resistant allele. And when we look at our legacy populations, those are the 2007 populations. With the exception of Kern County, you can see that few individuals had those phenylalanine alleles present. But when we look at the modern population, almost every single individual, with the exception of Coachella Valley, has some sort of resistant allele in their genome. With this, I also want to point out that the instances of heterozygotes has increased over time. If you look in each county, you see that in the legacy populations, there were low or no occurrences of heterozygotes. And in our modern populations, these heterozygotes definitely make up a significant portion of the population. And that's so important because heterozygotes and homozygotes imply certain things about the survivability of these populations and these individuals. This is a CART model made by Billy Mortola in 2023, and it tells us things about heterozygote survivability. I'll walk us through this a little bit more. Down on the bottom left here, we have all individuals that are homozygous for the wild type or possibly have a serine allele in them. This CART model did not differentiate for serine. Their survival probability was 7%. Moving on, we have F heterozygotes. Again, this is a mix of phenylalanine serine and phenylalanine leucine, and their survivability was an 18% chance of survival. Next, we have F homozygotes that have a 76% chance of survival when exposed to pyrethroids, and that's pretty concerning implications for what we've seen here. And this all, the CART model and our findings are not surprising when we look at what's been happening phenotypically and the implications. So what I have up here on the right are two bottle bioassays from the Yolo County. That is one of our Northern California sites. In 2012, you see a pretty good, we're happy with it, bottle bioassay where that light blue population, that's our colony, died as we expected it to. And the wild colony or wild caught mosquitoes die within the bottle bioassay and reach near 100% mortality. We have a very different story in 2022. In 2022, this bottle bioassay shows that our susceptible colony acts exactly as you would expect it. However, the wild population didn't reach even 75% mortality, which is well within the diagnostic criteria of a resistant population. So where do we go from here? Is it possible to recover susceptibility? The answer is it's really difficult, but I suppose not completely impossible in some populations, as heterozygotes with that wild type do exist. We talked earlier about how this mutation comes at a fitness cost. That's those weaker depolarizations. However, this fitness cost does now outweigh the benefit of not dying when exposed to pyrethroids. And this inheritance pattern leads to susceptible alleles, unfortunately, not persisting. And this is supported not only by our findings in these populations, where you see in two of our counties, Kern and Sutter, that there were no wild type alleles left in the population. That is also supported by the CART model when we can translate these kind of statistics to what we would expect phenotypically in these populations. So moving on from here, we would like to investigate the relatedness of resistant populations. We found it really interesting that Southern California and Northern California told such different stories, and it sparks questions on, was this something that existed in Southern California and, for lack of better terms, spread up to Northern California, or were these independent events? We would also like to look at the survivability of these populations, specifically looking at what does that fitness cost really mean to these mosquitoes. And finally, as a larger community, it's easier said than done, but the way we fix this is doing better. But what does that even mean? It has a lot of challenges. That's continuing to implement IPM and IVM, which includes education and novel methods and pesticide rotation. And with that, I think I have reached time, and that's all I have. So I need to acknowledge all of the districts that sent us mosquitoes throughout this study. Thank you very much. Your hard work is seen. University of the Pacific and PacVec, of course, for funding and a place to do grad school, and specifically the Sacramento Mosquito and Vector Control District, because I am giving this presentation instead of doing my work right now. Thank you very much. Are there any questions?
Okay, thank you, Anna. If there are any questions for Anna, then please raise your hand or post it in the chat. I'm not seeing any raised hand, or just speak up if you've got a question that you'd like to ask.
Hi, Anna. Really nice talk. I do have a quick question. I was really impressed with the historical data that you presented on the historic insecticide usage, particularly related to agriculture and pest control. I think that's something that we'd really like to get a good idea of here in the Northeast. So I was just curious how you kind of were able to get that historical data.
Yeah, the California Department of Pesticide Regulation maintains records of all of the reports to it, all the way back to, I believe, when it was founded. And you just have to request those records from CDPR. I'm not sure where you are or if there's an analogous organization, but the way it was CDPR, the way it worked, it was CSV data that I then had to tease out in R and sort using their manuals because everything was encoded, which was kind of a pain, but the data was there. And I wouldn't be surprised if similar data was available through your analogous agency.
Awesome. Thank you.
Yeah, I have a couple of questions. One is Anna, what is known about sort of dispersal of these Culex species? And do they know where the origin of this mutation arose, and how it might have spread out from one location, or do they think it was multiple sorts of multiple times that it's arisen multiple times across different populations?
That's what I would really like to know too. So that's definitely a question that came up once we saw our data, particularly with Northern California and Southern California looking so different. I don't believe we do know the origin of the KDR mutation. But the team in lab right now is doing a little bit of teasing into the genetic relatedness of these mosquitoes and trying to determine if that multiple event versus single origin theory, which one is more likely to be true.
Okay, and then another question I had was that it looked like the Coachella data were a bit different from other data that you'd collected in terms of the preservation of the sort of the wild type. Do you think that's due to the sort of the type of agriculture that they do down there in comparison to other parts of the state where it's a sort of more traditional kinds of agriculture that they have?
Yeah, that's a great question because Coachella Valley does look so different. For that, I'm gonna click block in my slides till I find this one. So Coachella Valley is down in Riverside. And what you can see here when normalized by acreage is that their main selective pressure was structural pest control. And that's an interesting definition within itself. And structural pest control is defined by CDPR as applications taking place within in structures. So I think that means like tenting for termites and stuff, which is not only non-target exposure, but would only place selective pressure on mosquitoes through residual insecticide presence. And besides that, Coachella faced very low selective pressure when compared to most other counties. What's interesting here is that it's a similar situation to Lake County, which is up at that top right. But Lake still saw that really strong shift. So I'm not exactly sure why we've seen that wild type persistence in Coachella, but that's definitely a little bit of why I really want to tease into that question of how much of a fitness cost does this mutation have, and kind of how much does that weigh against the benefit of resistance, especially in these populations where they're not facing very hard selective pressure.
Yeah, I mean, there is agriculture down in Coachella. It's just probably not the same kinds of, they're probably not using the same kinds of products as they are in other places.
Yeah, this graph importantly does only highlight pyrethroid application. There's nothing about organophosphate exposure here, which is certainly another interesting thing to look at. We also didn't look at other mechanisms of resistance within these mosquitoes. We focused on KDR, but that's not to say that these mosquitoes didn't have other mechanisms going on within them.
Okay, and Chris Barker had a question about Lake County, but he posted a second comment just to say that you anticipated his question and answered it. So thanks, Anna, for great work.
Thanks, Chris.
Any other questions for Anna? If not, I think, Anna, you can stop screen sharing and we'll move on to our next speaker.
Great, thank you.
Thank you, Anna. Well done. Thanks, Scott. And our next speaker is Aine Lehane. Aine is a technical advisor with the Northeast Regional Center of Excellence in Vector-Borne Diseases. So this is a cross-fertilization across our two different centers of excellence for vector-borne diseases. So Aine is going to talk about the cross-COE Center of Excellence in Insecticide Resistance Working Group that she leads. Take it away, Aine.
Thanks, Neil, and thanks for inviting me to the PacVec seminar. Happy to be here. All right, hopefully everybody can see my slides here. Like Neil mentioned, I work with NEVBD, the Northeast Center for Vector-Borne Disease, and have been leading this cross-COE insecticide resistance working group for about the past 2 years now. So I just wanted to start with kind of introducing the working group. So you can see here in this graphic that we have representation pretty much across the US as well as in Puerto Rico. We have members who work in local mosquito control, as well as various members, various branches of the federal government. So USDA, CDC, DOD. We have a member who works at a State Department of Health, various participants who work in academia, as well as those who are associated with a COE or tech. And we recently set up a website. So if you are interested in kind of reading more about who our members are and our different projects, feel free to check that out through that link in the top right or the QR code. But yeah, basically the working group is really just wanting to address some of these gaps in our knowledge that exist that relate to insecticide resistance in mosquito populations. And hopefully answering some of these questions and kind of disseminating that throughout our community and to our partners. So for the talk today, I'm just going to focus on 3 main projects that we are currently working on at the moment. The first is looking at modifying the CDC bottle bioassay analysis to capture more quantitative data. And then we'll also talk about a second project, which is comparing using formulated product versus technical grade active ingredient in the CDC bottle bioassay. And then finally, a third project is putting together this best practice guidance document for evaluating ultra low volume applications of insecticides for adult mosquito control. So I'll start with this kind of new way of analyzing the CDC bottle bioassay. This has really been led by Ed Norris out of USDA. He's really been spearheading this, and a lot of the data that he's used for this analysis was provided by Casey Parker Crockett as well as Sarah Wheeler. So just want to give some credit there to those folks. So just want to start out with mentioning the CDC bottle bioassay. This is a really widely used assay, and so there's a lot of kind of historical data that we can kind of make use of that's come out of the use of this particular assay. But there is this kind of limited capacity to quantify the degree of resistance, even though there is a lot of existing data. So this is where this kind of new analysis method comes in, really trying to generate a more fine scale interpretation of resistance levels and then compare that to field cage trial data. So Ed did a survival curve analysis using the Kaplan-Meier analysis. And just to back up for a second, the CDC bottle bioassay protocol states that in part of that assay, the mortality is monitored over time. So you have these time points where mortality is measured. And so we can use this survival curve analysis to generate a statistical estimate. So in this case, the median lethal time. And we can use that to compare levels of resistance across different mosquito populations. So just to kind of lay out the objectives of this study, one, it's really to understand if the current data sets that we have of the CDC bottle bioassay, could those be used to generate survival curves? And then secondly, can we determine if the survival curve analysis generates consistent statistical outputs that can be compared? And then finally, we want to understand if we can compare the survival curve statistics. So again, the median lethal time, can we compare that to field cage trial data and determine if there's a correlation between the two? So this is just an example of some of the data that Ed used in this analysis. So it included multiple different mosquito species. You can see those listed on the far right-hand column. And we also had a variety of different active ingredients included in the data set. And then only data sets that had a corresponding field cage trial data set were included so that we could pair the two. And then we didn't include any data where synergists were included. And so then there was just this concern of, are the results consistent across replicates? So this data was provided by Sarah Wheeler. And, oops, sorry, let's go back here. We can see on the bottom left, a susceptible mosquito population was treated with deltamethrin using the bottle bioassay. And then on the right, we can see that this same population was treated with deltamethrin again 2 months later. And when we look at the mean estimate between the two, the results are very consistent. So from that, we just kind of were able to conclude that there are consistent results between replicates of the same mosquito population. So next, the next step was to correlate that Kaplan-Meier survival analysis data to our field cage trial data. And so with the field cage trial data, we really wanted to align those data sets. So we selected data sets where the distance from the spray path was 300 feet and the time after spraying was 12 hours. And then, like I mentioned before, the data sets were paired. So we had bottle bioassay data paired with field cage trial data using the same ingredient. So just to kind of give an example of this, in the field cage trial, if DeltaGuard was used, the main active ingredient in DeltaGuard is deltamethrin. So in the corresponding bottle bioassay, deltamethrin was the active ingredient that was used. And so then from there, we were able to compare mortality in the field cage trial data sets to the median lethal time in the bottle. So when we look at these results, we can see that there's a strong correlation between the median lethal time and the percent mortality in the field cage trials. So we were pretty happy with those pretty strong correlation results. And then Ed is very thorough and followed this up by also looking at a correlation between the resistance category and the percent mortality in field cage trials. So in using the bottle bioassay, we can determine if the population is susceptible, developing resistance, or is resistant. So with those three categories, we see a pretty weak correlation with mortality in the field cage trial. So with this particular data set and this set of analyses, we kind of concluded that the CDC bottle bioassay categories aren't sufficient for correlating field cage trial data. So just to kind of summarize the main conclusions from this first project, we saw that repeated survival analysis showed that there was consistent mean lethal times for matched populations tested many months apart. We saw that there was a strong correlation between the mean lethal time and the mortality in field cage trials. Like I just mentioned, we saw that there was a weak correlation between the resistance index and the field cage trial mortality. It is possible that using a different distance, different time points, that you might be able to get a stronger correlation there. But at least with the data that we had to work with, we didn't find a strong correlation. And then kind of overall, pretty much looks like the Kaplan-Meier survival analysis might be an appropriate alternative analysis method to correlate this CDC bottle bioassay data to field cage trial data. And Ed is continuing to work on this along with others in the working group. So some future directions are to get additional data sets, really try to bring in more mosquito species, looking at additional active ingredients and seeing if this correlation holds. We'd also be interested in looking at performing a correlation with the CDC bottle bioassay data using formulated product as opposed to using just the active ingredient, as well as looking at evaluating correlation of the survival curve analysis to topical application data. So Ed is actually in the process of receiving some topical application data at the moment from some collaborators. So stay tuned on that. Hopefully we'll have some nice results from that as well. All right, so next I'm going to switch gears here and talk about a second project that multiple members of the working group are currently involved in. And this is looking at formulated product versus technical grade active ingredient in the CDC bottle bioassay. So just to start with some quick background information here, like I mentioned before, the CDC bottle bioassay is very widely used for assessing insecticide resistance in mosquito populations. And a lot of programs and mosquito control agencies have adopted this bioassay, but as they've adopted it, some modifications have been made. And so one of the most frequent modifications that we hear about is substituting technical grade active ingredient, which is what the protocol calls for, to using formulated product. And so formulated product is the product that's used in the field. So that contains the active ingredient as well as some additional kind of inert ingredients. So using that in the bottle bioassay. And programs are tending to use formulated product in the bottle bioassay. I think maybe because it's readily available to operational programs. And there might also be this perception that using the formulated product might give you more relevant kind of results to know how efficacious the product will be in the field. But that comes with challenges as well. If you're just using formulated product in the bioassay, that might kind of obscure what the true resistance is in the mosquito population. And there's also no kind of set protocol or calibration on how to conduct sort of a modified assay with formulated product. So the objective here for this project is to compare mortality of mosquito populations using 3 different assay methods. So the first would be to run a standard CDC bottle bioassay. This is using technical grade active ingredient. That's what the protocol calls for. The second would be to run a modified assay, so using formulated product at a concentration that matches what the concentration is in the technical grade assay. And then the third is doing another modified assay with formulated product at a concentration that's kind of reflective of what would be applied in the field. So you can see in this chart on the bottom, if we look at the first row, if we are using etofenprox as an example here, that's the active ingredient. The formulated product that will be used is Cenevex, and then we see the varying concentrations of the product in the bottle, depending on the assay type. Okay, and then in terms of methods, the standard protocol is being followed for all of the treatment groups, kind of minus what product is actually going into the bottles. And mortality is being recorded every 5 minutes over the course of this 2-hour assay, and then conducting 3 true replicates each. So in this chart, we can see the 3 different species that will be tested. So we have Aedes aegypti, 2 populations of Culex quinx, and 2 populations of Culex pipiens. These are all susceptible populations and are being tested by various members of the working group. So this is an ongoing project, so I just have a few preliminary results to share today, but hopefully we'll have more information to share soon. But this is looking at a susceptible Culex pipiens population that was exposed to deltamethrin. This chart is similar to what Anna was showing with her bottle bioassay data. So we have time on the x-axis and mortality on the y-axis. And the red line represents the technical grade kind of standard protocol of using the active ingredient in the bottle bioassay. And the blue line shows the formulated product assay, and then the green line shows the formulated product at a mid-label rate. And so when we compared these, we saw that there wasn't a significant difference between the formulated product assays, but there was a significant difference in terms of mortality between the technical grade product and both formulated product assays. And this was the case as well when we used the same susceptible Culex pipiens population and exposed them to etofenprox. So again, we saw that there was a significant difference in the technical grade active ingredient assay compared to the 2 formulated product assays. So still a lot more to do on this project, but just some preliminary findings. It does look like there is a significant difference in what we're seeing between using formulated product in the bottle bioassay versus using the active ingredient. So this just really kind of indicates to us that formulated products shouldn't be used in place of the technical grade active ingredient in the CDC bottle bioassay. And that there definitely is a need for developing a formulated product assay with a protocol that kind of has diagnostic criteria for each of these formulated products that could be used in the bottle. But that's kind of down the road. The next steps here with this project is to repeat replicates of this experiment with susceptible Aedes aegypti, and then also with Culex quinx. So once those get started, it will be interesting to see if we see the same outcome with those two different mosquito species. Okay, and then just to move on to this third and final project that the working group has been tackling. This is related to putting together a guidance document on kind of how to conduct a cage field trial. So to start off with some background here, again, CDC bottle bioassay, that's often a really common first step that programs or agencies use to determine if there is some level of insecticide resistance in their mosquito population of interest. But cage field trials can really show how effective a product will be at controlling your mosquito population in the field. So there's definitely value to kind of going out into the field and doing these cage trials. But the main problem that our working group identified is that defined methodologies for conducting these field cage trials are not widely available. So that kind of leads us to our main objective, and really the purpose of putting together this document is to put some standardized guidance out there on how to best conduct a cage field trial. And the goal is to really ultimately work with AMCA to get this published as a best practice guidance document on their website. So next, I'm just going to cover some of the main topic areas that we have outlined in this guidance document. This is still currently being drafted, so there are additional topic areas that we want to add, but I'll just kind of walk you through what we have so far. And so I'll start with the study design, and we focus on ground-based applications first. So this would be doing ultra-low volume sprays via a truck-mounted sprayer. So we kind of provide guidance on that, as well as talking about considerations in terms of when to do this in terms of timing and what weather and wind considerations exist as well. We also provide guidance on selecting an appropriate site for conducting one of these trials, as well as how to best design your transect. We then follow that up with how to design an aerial application and kind of provide some comments on what are the differences between doing a ground application versus an aerial application. We also have some recommendations on doing that block design for your aerial spray, as well as some ideal site selection considerations. And then we also have considerations in terms of the spray height, how to calibrate the swath width, and thinking about wind speed and what impact that will have on the trial as well. We have a whole section on equipment. There are a lot of parts involved in these trials, and I think that can be a hurdle sometimes for folks who are trying to get started with a field cage trial. So we have a list of the equipment that's needed to run a trial like this. We have a section on the cage types and what the options are, instructions on how to make them. We also plan to include resources in terms of where to kind of source the raw ingredients for making cages, all the way up to where you could purchase the cages. So kind of having different levels of options for folks in terms of the materials that they would like to buy. We talk about mesh size, as well as using tripods, slides, rods, and impingers. We have also a section on mosquito selection. So really kind of talking about why a susceptible mosquito population is needed and why they should be included in a cage trial. We also talk about the use of females only versus using both males and females. And then we also plan to provide methods and kind of a protocol for how to collect wild mosquitoes to use in your cage trial. We also have a section on droplet analysis. So there's recommendations on when to use impingers and when to have those running during the trial. But then we also have a pretty extensive section in terms of protocols for how do you analyze this droplet data, depending on what materials you have and what software is available to you. So we have protocols for using drop vision, A-drop, as well as manual analysis. We also plan to have a section on data recording as well as data analysis and interpretation. This is still very much in draft mode, but we plan to have a downloadable data collection sheet just to make it easy for folks if that's something that folks would like to have. We'll include that in our guidance document, as well as recommendations on appropriate analysis methods, and then kind of how do you interpret that data as well. And then finally, we also are planning to have a section on troubleshooting. So anyone who has done field work or done field cage trials knows that, issues just crop up when you're out in the field. And so we were thinking it could be helpful to kind of preemptively address some of these common issues that tend to come up, be that with equipment in the field, kind of some common issues there, how to maintain your mosquitoes prior to use in the trial, what to do if you experience control mortality. We've also kind of we've gotten input on this that weather can also be really problematic. So kind of addressing some of those issues in the guidance document is part of the plan as well. So with that, I just want to kind of give credit to every member of the working group. It's a really dedicated group, and I've been so impressed with sort of the work and the time that people have been willing to put into these projects. And I think folks are really motivated to kind of answer some of these gaps in our knowledge that I mentioned at the beginning, and kind of hopefully be able to provide resources to folks and answer more questions on insecticide resistance. But yeah, with that, I'm happy to take any questions.
Thank you, Anya. If anybody has a question, please either raise your hand or post it in the chat. Let's see, I'm not seeing any.
Neil, I have one. I was curious, Anya, is a really nice overview of what the working group has been working on. And I know one of the challenges with formulated products is just the difference in space sprays versus residual sprays. And there are pretty good formulated product assays for residual where you've got a surface and you can apply to the surface. But I was wondering if you could just sort of walk through how you think about a space spray, like an ultra low volume spray, and how to translate that into exposure through the cuticle, I presume.
Yeah, that is a great question. And I wish Casey Parker Crockett was on the call, because she worked some magic in terms of calculating the concentration that should be in the bottle that would correlate to the concentration that would be sprayed out in the field. And so I wasn't part of how she determined that, unfortunately. But I can find out for you. But yeah, I know that she kind of wanted it to be as accurate as possible to kind of reflect what is sprayed in the field.
Yeah, I'd just be curious about whether it's like essentially mimicking one droplet contacting the mosquito, or is it something else?
Yeah, and I think that's kind of a broader issue with the CDC bottle bioassay, is just in terms of contact that the mosquitoes make when they're in the bottle. You can't really know exactly. And I think our group anyway at NEVBD has been thinking about the value of doing topical assays, because in that case, you can know exactly the amount and concentration that you can apply directly to the cuticle. So I guess this is sort of a broader concern with the CDC bottle bioassay.
Okay, thank you, Anya. Any other questions? Okay, I'm not seeing any. So I'd like to thank both of our speakers today, Anna and Anya, for excellent talks. Oh, there is a question down here in the chat. It just popped up. Katerina Stokes says, we equated the size of the cage to the size of the bottle, but mode of exposure is definitely different. I think that was is that from Sarah Wheeler? Okay, any other questions or comments for the speakers today? So I'd like to thank both of our speakers again today, Anna and Anya, for excellent talks on the theme of insecticide resistance, which is a very important one in the mosquito control world. And I would like to remind everybody that our next talk or seminar will be on May 13th. Every couple of weeks for the next month or two. And we'll look forward to seeing you there. Please join me in giving our two speakers today a round of applause, or at least a clapping emoji to show your appreciation. Both of them did an excellent job. And thank you very much. And we'll see you on May 13th.